So first, I would like to thank the organizers for having given me the opportunity to present our study. The study was carried out in the group of Professor Joel Stavans from the Weizmann Institute of Science. As you probably know, circadian clocks can play a key role in many biological processes. Circadian clocks from unicellular organisms to an animal may increase fitness by temporarily aligning biological processes with day and night cycles. A lot has been learned about unicellular cyanobacteria in the context of circadian clock in the unicellular organism Synechococcus. And in this organism, the core circuit controlled post-translationally by three proteins, Chi-A, Chi-B, and Chi-C. Chi-C has two phosphorylation sites at serine and threonine, and Chi-C can be in one of the four phosphorylation states: unphosphorylated, phosphorylated at T, both at S and T, or only on S. In addition, Chi-C is regulated by Chi-A and Chi-B in a feedback loop configuration. Chi-C transduced its output signals down to downstream genes via the master regulator transcription factor, RPA. Some of the input signals are the ATP-ADP ratio and the redox state of the cells. So we wanted to study how do circadian clocks behave in multicellular cyanobacteria. In order to address this question, we choose to study Anabaena. This is a wonderful one-dimensional filamentous cyanobacteria organism. As you can see in this picture, this cell consists many cells, can be hundreds of cells. There is cell-to-cell communication between the cells, and therefore that suggests clock coupling and even synchronization. So not much is known about circadian clocks in Anabaena. The only study that was published about circadian clock oscillation was done at the level of bulk study by DNA array measurements. They have shown that about 100 genes exhibited oscillatory behavior, but none of the Chi genes of Anabaena displayed high amplitude expression returns, as opposed to Synechococcus. So we wanted to study a clock control gene in which have high oscillatory behavior. We choose to study PEC-B, which codes for a subunit of photosynthetic pigment. And this gene PEC-B, we study that by following the expression of PEC-B fusion to the promoter of PEC-B fused to GFP, a translational fusion at a single cell level in real time under fluorescence microscope. So using we also have phase contrast images. We segmented the cell and measured the mean fluorescence intensities of each individual cell along the filament. So having this strain, we took movie. So here I show you a movie a typical movie that we took in the lab. At the left, in red, you can see the autofluorescence pigments in Anabaena. In the right, the expression of PEC-B from its promoter. So this movie was taken for about 5 days, every 30 minutes, in a gel pad-based device. The cells were grown under constant light condition, which suggests the autonomous behavior of the clock. And as you can see, synchronization and spatial coherence maintained despite cell-cell variability. So having these movies allows us to measure the clocks at the individual level, at the individual cell level, and to measure their lineages. So here I show you two cells and several generations. You can see from this figure that the phase is inherited upon cell division and that the lineage preserves ancestor phase for several cycles. We can also measure the average fluorescence intensities of the filaments, and you can see here in pink that oscillation maintains several generations. However, when we deleted the clock genes, as shown in white, the oscillatory behavior was abolished. Note that the oscillations are abolished or there is no autofluorescence pigment oscillation in the wild-type background strain. So in order to study the special correlation along the filament of PEC-B, we measured the special autocorrelation of the fluorescence along the filament by use the autocorrelation function. So this function relates the fluorescence of cells with that of its end neighbor. As seen here in this graph in blue, we measure the autocorrelation function of the wild-type background, and we find that it decays in about 5 cells. So we can this correlation can be due to the inheritance after cell division or due to cell-cell communication via septal proteins. So in an abionaite, it was published that septal proteins such FAST-C and D may transfer small sugars and metabolites, but not core clock components, and therefore we developed a strain in which we deleted this septal protein, and as shown here in green, you can see that the autocorrelation function decay in a length of about 2 cells. Note that similar decay was observed in the Chi deleted background as well. So deletion of clock genes or septal proteins involved in sugar and metabolite transport reduce septal coherence. In addition, for comparison, we show in pink the correlation that would expect solely from inheritance after cell division obtained from stimulation. And as you can see, the inheritance after cell division accounts partially for correlations. In addition, we wanted to measure the synchronization in the filaments. So in order to do that, we calculate the synchronization index as suggested previously in the literature. The synchronization index is defined as the ratio of the standard deviation of the mean fluorescence intensities of the cells with time, divided to the standard deviation of the intensity of cells along filament, averaged over all cells. So this index is varied between 0, which is unsynchronized, to full synchronization, which is 1. So at left, you can see here, for example, the filament. We follow along the synchronization or along the growth of the cells, but we choose cells that are uncorrelated. So we took enough distance, at least 10 cells, between the cells that we choose to analyze. And when they grow, you see that this distance is increased. So we found that the wild type background, this index is about 0.85, which is a quite high value. However, when we deleted the septal proteins, in this background, we found that the value is significantly smaller. So disrupting cell-cell communication reduces synchronization. So to sum up, today I showed you that circadian clocks in anabaena are coupled via cell-cell communication, that oscillations are highly synchronized and spatially coherent, and for a stochastic model that includes demographic noise in phosphorylation state, see our upcoming paper. So I would like to thank the people that contribute to this study. This study was done in a group of Professor Gerald Stavans with collaboration with the group of professors Enrique Flores and Antonia Herrero, and the group of Duccio Fanelli from Florence. And I would like to thank you for your attention, and I will have a question now.
Thanks a lot, Rinat. Maybe I'll start with my own question. So there are situations in your beautiful movie where the sort of the filament folds and you have contact between faraway places. So I'm wondering if synchronization is only along the line, or even just adjacencies could somehow synchronize.
So the cells which are touching each other are in very long distance. The coherence is quite, we find it along the filament, but there is no cell-cell communication between these cells. It's just due to some sugars that there is contact, but there is no, we assume that there is no real transport or things that are correlate. So it's actually via the contacts between the cells, meaning channels like the septal protein. It might be others, of course.
So Jason Hughes asks, how fast do these cells migrate, or how much motility do the filaments have, as cells moving in and out around each other could alter and impact the spatial correlation? So I guess kind of related to what we were just talking about.
Okay, so the cells are just, it's like it's a filament, so it's like, there is no, they are contact like that. It's a real bonafide, so it's a filament that all the cells are connected. And therefore, the motion is due to their growth, because each time in between the cells are grow and they just become longer and longer. So it's like many cells that are in a train that every time it's split in the middle. So this is the reason that you see that, but actually the cells are in a gel pad that is between two in slide, and we keep the focus for several days. So the migration, there is no real migration, it's just the cell division. What we see. It's due to cell division, the growth of the filaments.
Right.
Okay, and maybe I'll ask one more question. Can you say something about the evolutionary advantage of these long filaments and sort of their sort of the limitations? Can they just continue to grow? Are they occasionally broken up into single cells or smaller filaments? Do they ever merge? Anything like in general about the biology of this weird organism?
Okay, so actually, if you know, Synechococcus is a single cell, but in the case of Anabaena, what is beautiful about there is no merging. Actually, what happened is that in this is very, it can be hundreds of cells. It can be very, very long. And with time, sometimes they break and they continue to grow, but they can be sometimes hundreds of cells. So it's like a very long filament. And the binary division is in E. coli. So they just divide each time, and then you have another, every 24 hours, you can see in average cell division. It's not so fast like in bacteria or in Synechococcus, so it's every 24 hours. So they have a longer cell cycle. Yeah.
Okay, so there's one comment from Arjun that got several votes, which is just a comment about how beautiful your movies are.